It is now my pleasure to introduce our next speaker, Beronda Montgomery. Beronda Montgomery is the Michigan State University Foundation Professor of Biochemistry and Molecular Biology and Microbiology and Molecular Genetics in the Department of Energy Plant Research Laboratory and Assistant Provost for Faculty Development and Research at Michigan State University. Professor Montgomery joined Michigan State University in 2004. Her laboratory investigates the mechanisms by which plants and cyanobacteria, which have limited mobility, monitor and adjust to changes in their environment. The ability of these organisms to adapt their patterns of growth and development to fluctuations in external environmental parameters increases their survival and maximizes their growth and productivity. Dr. Montgomery also conducts scholarship and training initiatives on effective research mentoring, research management, and academic leadership, and has a special focus on mentoring diverse students and junior scientists as well as faculty development. Dr. Montgomery's scholarly efforts have been recognized by receipt of a National Science Foundation Career Award, being selected as a finalist in the 2014 Howard Hughes Medical Institute Professors Competition, being a 2015 Michigan State University nominee for the Council for Advancement and Support of Education U.S. Professor of the Year Award, and being an ASM Distinguished Lecturer from 2017 to 2019. Please join me in welcoming Professor Montgomery as she delivers her presentation, Seeing the Light and Shaping Up, Color Vision and Developmental Acclimation in Cyanobacteria.
Good evening. I think I'm actually going to want a song every time I enter a room now. That was nice. So Victor Rita is here. He can make note of that for microbiology faculty meetings. Any of you who know me well know that a captive audience is my dream. So I want to thank you for being here and also thank the committee for allowing me to talk about my work. As you heard, I work with plants, cyanobacteria, and people, really trying to understand how organisms know what they are, what they should be doing, and engaging with the environment. And tonight I'm going to tell you about some of our work with cyanobacteria. So this organism, if you've ever heard me talk about it before, I often talk about it as the second most beautiful organism on the planet. The first is 15 years old and is in charge of my elder care. So he gets number one always. This organism is beautiful because of its flexibility and adaptability. And so what you're seeing here are two images, one reddish in color, one blue-greenish. These are the same organism. In one case at the top of the slide, the organism has been grown in green light. It's reddish in color because it has about 40% of its total soluble proteins that are green light absorbing proteins linked to photosynthesis, so it reflects red to the eye. That same organism, when you grow it in red light, is blue-green in color. Cyanobacteria derived the name. And that's because in this case, the organism perceives that the external light is red and makes those 40% of the total soluble proteins linked to light absorption as red light absorbing proteins. And so this is a photoreversible process whereby the organism is able to tune its pigmentation to the available external light to optimize its ability to absorb light for photosynthesis. What you also see there in a micrograph from a 1973 paper by Bennett and Bogorad is that the organism also has changes in its cell shape. So in one condition in green light, it has longer filaments. This is a filamentous cyanobacterium, and each of the cells is rod-shaped and longer. In red light conditions, the filaments are shorter and the cells are more spherical. And so this organism, whose name is Firmella diplosyphon, at least in the US. In Europe is Calothrix species PCC 7601. That divide is a talk for a different day. Today we're going to talk about this organism as Firmella diplosyphon. This morphology is also photoreversible. And it makes sense to us that an organism would tune its photosynthetic pigmentation to external light, but we also want to understand why it's investing energy in tuning its cellular morphology. Now, although this organism has this beautiful phenotype, this is also very important in a natural context. So these are aquatic organisms, and at the surface of the water, there's lots of red light available. And so at the surface of the water, there are other photosynthetic organisms that use red light for photosynthesis. But as shown here, red light also just doesn't penetrate water as well. So deeper in the water column, there's still blue and green light that's available. So organisms such as Frimella that are able to adapt their pigmentation between red and green light actually have an expanded niche available for their growth compared to those that can only use red light for photosynthesis, or some marine organisms that primarily use blue light. And in addition to these changes in the available wavelengths of light, there's also a difference in how bright the light is at the surface versus how dim it is. And we look at some of all of these characteristics, although today I will focus primarily on wavelengths of light. What we know about those shapes is that the smaller spherical cells are at the surface, whereas the elongated cells are deeper in the water column. And that helped drive some of the hypotheses that we made about why the organism may be investing in optimizing its shape under different conditions. So in addition to being able to see this as you saw it on the plate that I showed you, if we do absorbance spectra, you can see that there are distinct pigments. And so here the color of the line is the color of light that the cell has been grown in. And so you'll see that there are peaks at about closer to the 400 nanometers and closer to 700 that are apparent in both red and green. Those are the chlorophylls that are at the center of the reaction center. These organisms have accessory light harvesting complexes, which look there like a fan on the right part of the slide. Those are phycobilisomes, and that's where these 40% of total soluble proteins are occurring. So you'll see that in green light, there's a unique peak at about 565, 570 that's labeled PE, phycoerythrin. This is the green light absorbing protein. And in red light, there's a distinct peak labeled PC, or phycocyanin. And so these are the distinct pigments that you can see purified in the glass tubes at the very far right of the slide that are bright blue for the phycocyanin, the red light absorbing protein, or pinkish to red for phycoerythrin. And so a number of years ago, a study was done to try to understand what regulates this photoreversible switch between these red light and green light specific proteins. And the way they did that was to do a mutant screen whereby you can grow cells in red or green light. These cells have a very nice property that they have stable insertion elements that you can induce to move under very defined conditions. And so we have a protocol under which we can treat the cells either with heat or cold under defined conditions. The insertion elements then move in the genome. Where one lands in the middle of the gene, there's no longer a functional gene product made, and you can assay to see if that has an impact on function. So after mutagenesis, we can plate the cells out, and under red light, we look for things that are not the wild type blue-green color, or under green light, we look for things that are different from the wild type red color. We isolate those, and then we're able to do a number of assays to define where the insertion element is, which gene has been disrupted, if there is a disruption in the phenotype. Such a study led to the identification of a mutant that was named Black BK14. It was black in color, you can see, and it was the 14th such isolate. We determined when you look at the spectral scan for those mutants at the bottom of the slide, unlike wild type that has the specific green light or red light peak, this mutant has all of the pigments all the time. So it's basically blind to external light. And when we identified which gene was disrupted, it was a photoreceptor, which was responsible for perceiving either red or green light and tuning the pigmentation. This mutant was named RCAE. This process, I failed to tell you, is named complementary chromatic acclimation, and this was a regulator for complementary chromatic acclimation, the 5th such gene, thus RCAE. When we look at the actual protein that is encoded by the gene, we found that it was a class of proteins that was already known from higher plants called phytochromes. These are photoreceptor proteins. They have an N-terminal photosensory domain that contains this chromophore binding domain shown in yellow or gold with the conserved cysteine residue. At this conserved cysteine residue, a chromophore is attached. In this case, it's a linear tetrapyrrole with 4 pyrrole rings. And between the 3rd and 4th, there is a double bond that on the protein perceiving light, there's a rotation around this double bond, which is enough of a change to cause a shape shift in the protein that then is activated. The activation occurs in the C-terminal domain, which is a histidine kinase, which means that it takes a phosphate from ATP, showing that it's been activated, and that initiates a phosphotransfer cascade that ultimately results in transcriptional regulation, tuning the pigmentation of these cells. So we were able to show that this gene was indeed the gene controlling this phenotype, because when we put, I'm going to go back one. When we put a plasmid only containing this gene into the mutant, we were able to recover the pigmentation back to wild type. So a single gene was responsible for disrupting that pigmentation phenotype in the mutant. One of the questions we had is, I told you before that both the pigmentation and the morphology are photoreversible processes. And so we wanted to know whether the same photoreceptor was controlling pigmentation as it was controlling the morphology. And since we had the mutant missing the RCAE, we could simply do microscopy to ask if the morphology was disrupted in this particular mutant. We also had to go back and show that in our hands, we could see the same thing, the red-green difference that they published in 1973, which was before the birth of the graduate student doing this work. Okay. So when we looked at those cells, we were able to see that, again, in green light, you have longer filaments. Each of the cells itself is longer and more rod-shaped. And in red light, we have a different and brighter red light, so we saw some stress here, but we did see spherical cells and shorter filaments. What was most interesting is that we were able to see that the mutant had a disruption in its phenotype, and that it was always stuck in a spherical shape. And so we have a single photoreceptor that's controlling both the pigmentation and the morphology, as we were able to determine also by complementing the mutant, although I don't show you the data for that today. So we know that we have a photoreceptor that's controlling pigmentation and also controlling morphology. This photoreceptor has to be working through some effectors. And what we know is that this is not a classic two-component phosphorelay, but it's a complex phosphorelay. A work of others, including David Kehoe and Arthur Grossman, were able to identify the effectors that function downstream of RCAE. And we were able to understand that there's a final DNA-binding transcriptional regulator that's phosphorylated, controlling the pigmentation genes. And in my lab, we've been asking which genes are controlled in the control of the morphology. And I'll tell you a little bit about that today. So the kinds of approaches that we generally take are to do mutant studies, which I've already shown you. And we largely do color screens where we get something that is not, wild type is either in the red or green form, depending on whether it's in red or green light. We get things that are stuck in one state, they're always red. We get cells stuck in the other state, always green. You can see here we get blue cells, we have gold cells, we have a chartreuse mutant. If you identify which gene is disrupted, you get to name the mutant, and the graduate student named it chartreuse. I learned a new word, a new color, okay? So in addition to the pigmentation screens, we also do a number of things like omics. We do proteomics and transcriptomics, where we're able to look at wild-type cells grown in two different conditions and ask which RNAs are being produced in one condition versus the other. We're also able to look at wild-type versus a mutant in the same condition and ask when a gene is contributing to differential regulation. Once we've identified genes that are of interest, we then go and do reverse genetics, where we're able by homologous recombination to inactivate a specific gene and ask which function it has. One other thing that has been particularly fruitful for us in terms of looking at the regulation of cellular morphology is to do what I call knowledge-based gene similarity analyses. So there are a number of bacteria in nature that are always spherical or always rod-shaped, and there are genes associated with the control of that morphology. These are sometimes referred to as morphogenes. And so we have simply asked the question whether these genes are present in the Frimella diplocyphene genome, and if they are, is their transcription controlled by light in a way that it would suggest that they are being effectors that are involved in the regulation of morphology. And so a number of genes that we identified, we then use again reverse genetics to knock those out by homologous recombination and confirm our hypotheses or not. And so one such gene is BOL-A. BOL-A is involved in cell shape regulation in a number of bacteria. Particularly in E. coli, it's known that during stationary phase, BOL-A is upregulated, and its upregulation is associated with a shift to spherical morphology. Excuse me. So we simply wanted to ask whether BOL-A was present in our genome first. And as you can see by this diagram, we found BOL-A in the genome. Interestingly, what we find across hundreds of cyanobacteria is that it's always sitting next to a glutaredoxin. And I'm not going to talk about that, but that was an interesting finding for us and others who have looked at a number of these bacterial systems. And so because the upregulation of Bol A is associated with spherical morphology, we asked whether in our cells, we know that the cells, when they're grown in green light are rod-shaped, in red light they're spherical, whether we could see Bol A differentially regulated. And compared to a control gene whose expression doesn't change very much between red and green light, it's not statistically significant, here I'm showing you images, although we did publish the QRT. I went for the fast visual because 25 minutes goes really quick. Okay. So what you can see is that compared to the control gene, we have an upregulation of Bol A RNA, messenger RNA, under red light conditions. So this suggests that we do see upregulation of Bol A under the conditions that we see spherical cells. So that's a correlation. We then wanted to test whether the Bol A in this system functions like E. coli. So we overexpressed it from a plasmid in wild type and asked whether its overexpression affected the morphology of cells. And what we identified, here I'm showing you wild type on top, DIC images, and then autofluorescence. And so those proteins, beautifully pigmented proteins, are autofluorescent. And we look at the autofluorescence simply to show that the cells are still alive. And basically what you can see is that, again, we see more rod-shaped cells in green light than we do in red light for wild type. But in the bottom half, when we overexpress the Bol A using its native promoter, you can see that we drive the shape of the cells, both in green light and red light, to more spherical. So it seems that overaccumulation of Bol A is associated with spherical morphology, and so we suggest that the upregulation under red light is driving in large part the spherical morphology that we see. So what's known about Bol A is that it's a transcriptional regulator, and it controls other genes. One set of genes that it controls are another set of morphogenes that have been looked at in bacteria, and these are bacterial actins known as MREB. And so it's known that Bol A can bind to in the genome and downregulate MREB expression in some systems. And so we simply asked whether MREB was present in Firmella and whether we see similar trends. And so we found MREB in Firmella in an operon shown here in gold with MREB C and D. And it's been shown in cyanobacteria already that MREB functions similarly to other cells. So in bacteria, it's relatively well known that MREB is associated with rod-shaped cells. So where MREB is present, the cells are usually rod-shaped. Where it's absent, not always, you don't see it quite frequently in spherical cells. And so it had been shown before, as shown in this part of the slide, that MREB in a different cyanobacterium that's always rod-shaped, if you mutate it, you lose that rod shape and you go towards spherical. So it seems that MREB is present in cyanobacteria and it functions similarly as in other bacterial systems. And so we ask, again, we have rod-shaped cells in green light. Do we see an upregulation of MREB under those conditions? So again here showing you qualitative RT, but having done the QRT in the paper that was published, I believe in Mold Micro, yeah, that's what it says on the slide, what we see is that MREB is indeed accumulating to higher levels under the conditions green light, that the cells are red in color and rod-shaped. And so it appears then that we are having light-dependent regulation of Bol A and MREB that is associated with the distinct morphologies that we see in Firmella. We have been able to show, and I won't show you the data here, that Bol A does bind and downregulate MREB expression in Firmella. What we wanted to know is whether this differential expression of Bol A that we see in red and green light is controlled by the photoreceptor RCAE that I've been showing you that controls pigmentation. And so we did quantitative RT-PCR to look at expression of Bol A in wild-type cells in green light versus red light, and in a mutant that's lacking the photoreceptor. And as you can see here again, we have a statistically significant upregulation in red light and wild type, but that's missing in the RCAE mutant. So RCAE seems to be functioning to drive the upregulation of Bol A in red light, which suppresses MREB expression, resulting in spherical morphology. In green light conditions, Bol A levels are lower, MREB expression is not significantly repressed, and then we see the rod-shaped morphology. And so what we've shown so far is that this photoreceptor, which we know is phosphorylated in red light, is working through some unknown effectors as of yet to drive expression of Bol A, resulting eventually in MRAB downregulation and spherical morphology. Conversely, in green light, Bol A levels are lower, MRAB expression then leads to this rod-shaped morphology. So we're doing some work to try to identify what those effectors are. I'm not going to talk about that today. But one of the other things I want to return to is that we're interested in why the cells are putting so much energy into regulating morphology. Again, we understand why you want to tune photosynthetic pigmentation. And there are a number of ways to go about this, but the approach we've taken is to do competition-based assays. And so basically, what we want to do are to take cells. The Bol A has the same pigments as does wild type. The only difference we really see is in the morphology. And so we want to put those cells in a direct head-to-head competition with wild type or other morphotypes and ask when and where does it matter that you have a certain morphology. And so basically we start cultures, the same number of cells, put them under a range of different environmental conditions, and at the end, by a number of different assays, we're able to ask who persists. And when and where does it matter that you have a certain morphology? So in the last few minutes that I have, I want to tell you the beginning of a story that I hope you'll go to the talk and a poster of my student to hear the end of the story. And so this is a story that came out of our transcriptomics. So in our transcriptomics, when we were looking at wild type cells grown in red and green light, and also comparing this to the photoreceptor mutant, we identified an interesting set of genes that were differentially regulated in the photoreceptor mutant compared to wild type. And we found a whole family of genes that were in an operon, CCM, for the carbon concentrating mechanism. And in cyanobacteria, they have a structure that allows them to concentrate carbon dioxide around rubisco to drive photosynthesis, as opposed to it being exposed to oxygen and having an unproductive reaction. And so what we identified is that this entire operon, the CCM operon, was largely upregulated. So here I'm showing you numbers for expression in the photoreceptor mutant compared to wild type. And so these genes were upregulated in the photoreceptor mutant. The rubisco itself in the middle in green, the large and small subunit, as well as the chaperone, were either not affected or downregulated. And then there's some independent genes that are part of the carbon concentrating mechanism that are not part of the central operon. So this suggested to us that the carbon concentrating mechanism in cyanobacteria was being affected and controlled by this photoreceptor. And the way these genes function is they make a structure that's known as the carboxysome. And so basically the carboxysome has rubisco in the center and a protein shell on the outside. Carbon dioxide in the form of carbonic acid is imported into the carboxysome. That's then converted by the carbonic anhydrase into carbon dioxide so that rubisco is in a carbon dioxide rich environment. Oxygen doesn't pass to the center, and so this is a way to concentrate the carbon. What we know is that the blue protein, the genes that are encoded in blue, encode proteins that make up the shell, the outside, and the rubisco is on the inside, with the purple and orange, CCM, M, and N, being the factors that nucleate the rubisco, and then the proteins are added on the outside, where you get a structure that looks very similar to a soccer ball with edges. Okay. So basically, this suggested that the carboxysomes, because the genes encoding them were misregulated, this suggested that we may see a phenotype if we actually looked at the carboxysomes. And if you do transmission electron microscopy, you can see a very nice six-sided structure of the carboxysomes in cyanobacteria. And we made a hypothesis that because you had about the same amount or less of the rubisco, but more of the proteins needed to package it, that we might have more carboxysomes that were smaller because you were trying to accommodate having more of this surface with the same or less amount of the volume that would go inside. And so when we looked at the carboxysomes, we saw a very nice structure. So when you look at wild type that's across the top for either green or red light, these white arrows are pointing to the carboxysomes. And In these images, you can't see a very big difference in the size or number for wild type. I'll show you numbers on the next slide. But what's very obvious to us is that in this photoreceptor mutant that I already showed you has spherical cells, we saw smaller carboxysomes, and they appear to be more numerous. So it appears that this photoreceptor is somehow controlling the carbon concentration mechanism through regulating the number and size of carboxysomes. The student who is here, Brandon Ronke, I used to say we, I've never measured a carboxysome. So the student, Brandon and others in the lab, measured the carboxysomes in terms of the size of the carboxysomes, as well as the number that we would see in a particular section. And what we were able to identify is that these are box plots showing the entire population. If you look at the first and third, this is wild type and red light versus green light. There are smaller, on this side we're looking at the diameter, so the size. There is a statistically significant difference between red and green light. So the photoreceptor is working in wild type cells to control the size. And the second and fourth are the size in the photoreceptor mutant. You can see that they're significantly smaller, which was obvious to see on the TEM. When we counted them, here we're looking at the carboxysome number per cell section, we also saw that there were indeed statistically significant higher numbers of the carboxysomes in the photoreceptor mutant. And so basically what we know now is that in addition to this photoreceptor controlling both cell shape and pigmentation, it's also controlling carboxysomes. And the question now is, what are the effectors that are being controlled by RCAE? And we do have some insights into the regulation of some of the structural proteins that Brandon is going to talk about in a talk tomorrow, as well as a poster on the following day. I was going to say either Friday or Saturday, but I have no idea which day it is. So it's tomorrow and then the next day that he'll be talking about that. Okay. And so I just wanted to end here by acknowledging the people who've actually done the work in the lab. The initial people working on this project were former postdocs, Sailendra Singh, Bhagni Patnaik, who's not even shown on the slide. Brandon has been leading a large part of this. It's a part of the Department of Energy funded project that we collaborate closely with Cheryl Kerfell, who is a structural biologist working on some of these proteins. And I also like to thank all of my funders, NSF, DOE, and the Michigan State Foundation. And I thank you for your time and your attention.